Hello, okay, guys, let's continue. So remember, I was telling you that C3 plants, their photosynthesis rate drop sharply if there is a lot of oxygen in the air. However, there is another type of plants called C4 plants, and I'm going to explain what this means. But their photosynthesis rate doesn't drop if the CO2 levels go higher, but instead of that, it remains the same. So let's kind of talk about that and see what is the difference between those two types of plants, C3 and C4. Now, the majority of plants on planet Earth, so about 95%, are C3 plants. C3-4, they exhibit something that's called a C4 pathway. Those are many of the plants that live in arid and dry environments. Many plants, such as majority of the grasses, like corn, sugarcane, and some other plants. So we're going to talk about in detail, about their biochemical cycle and what is happening. But that what enables them to do this very special biochemical cycle is something that's called Kranz anatomy. Kranz, this is a German word for ring. So they have this ring-like structure where you see inside the leaves, where vascular bundle is inside right here, and it is surrounded by bundle sheet cells. And then around that are the mesophyll cells. Something like that you can see in C3 plants right here. But of course there is, so this is a cross section of the leaf of the C4 dicot. But what is the difference is when we start looking at what happens where and which organelles are present. So, for example, in the C3 plants, only mesophyll cells have chloroplasts, so then they have rubisco, and then the Calvin cycle happens there. In bundle sheet cells, since there are no chloroplasts, there is no rubisco and no Calvin cycle. Now, when we look at the C4 plants, what we see is that both mesophyll and bundle sheet cell have rubisco. However, there is no rubisco in the mesophyll and then no Calvin cycle. So rubisco and the Calvin cycle are located on bundle sheet cell. And now this is kind of strange because photosystem 1 and photosystem 2 are present in the mesophyll and only photosystem 1 in the bundle sheet. So, let's try to explain what is going on. But before that, just one thing. So when carbon dioxide is mixed with water, then you have a carbonic acid, and then carbonic acid can very fast be dissociated into bicarbonates. And this is called the carbonate equilibrium. So this is one thing that we can have to remember because this is what is going to happen in the C4 cycle. Okay, so here is the mesophyll cell where in C3 plants, this is where the Calvin cycle is going to happen. However, in the C4 cycle, here is the Calvin cycle happening in the bundle sheet cell. So the thing that is fixing carbon dioxide, it is going to be, attached to water and then be dissociated, through carbonic acid into bicarbonate. So it is this bicarbonate that is going to be fixed to this new compound that we are mentioning called PAP carboxylase or phosphoenol pyruvate carboxylase. And when that happens, PAP carboxylase has 3 carbons, And now that is going to be converted into oxyl acetate. So now here you see this is kind of the first stable product, and it has 4 carbons, okay? So that's why this is called a C4 cycle. There are 4 carbons in oxyl acetate compared to C3, where you have 3PG. So the first stable sugar has 3 carbons.